Hello and welcome to this Chemistry World webinar, Emerging Routes to New Therapeutics, highlights from winning Horizon Prize teams. I'm Natalie Starkey, Chemistry World Science Media Producer, and I'm here with you for the next hour, where we'll be meeting some of the recipients of the RSC's Horizon Prize. If you'd like to use closed captions today, or English subtitles, in other words, in the webinar, then you can enable those using the CC button, which should be found in the bottom right-hand corner of your screen, depending on what sort of device you're joining us from. And I'd just like to point out that this webinar is also being recorded, so if you miss anything today, or you just want to watch again, then please look out in your inboxes for a link to the recording in the next few days. Now, as I said, today we're looking at some of the science awarded the RSC Horizon Prize. These prizes are the newest addition to the RSC Prize family, launched in late 2020 as an important part of the RSC's journey to make the ways scientific excellence in our community is rewarded and recognised fit for the way science is carried out today. The RSC's Horizon Prizes are currently open for nominations and will close on the 14th of January 2025 at 1700 GMT. The prizes are awarded to groups, teams and collaborations of any form or size and from any sector. The prizes recognise recent discoveries, breakthroughs and advances in the chemical sciences. Each and every individual who contributes to the prize-winning work is recognised as part of the prize. We've just shared a link to some of the pages on the RSC site that you can check out later. So please do have a look if you're interested in looking at the nominations there. You can also find out all the past projects that have won prizes, and there's a lot of material on there, so do take a look at that later. Now, I don't want to take up too much more time before handing over to our brilliant speakers today, but I wanted to let you know that, as with all Chemistry World webinars, this session is interactive, which means you can ask questions about any aspect of what's covered in today's event. Please use the questions box to send in your questions as soon as they come to mind, and I'll be able to put those to our panel of guests in the live Q&A towards the end of the webinar. We also have Ria from the RSC Horizon Prize team in the background, so if you have specific questions about the Horizon Prize or any other prizes, then please do send those in as well, and we can see if she can send you back a response today. But without further ado, I'd like to introduce our speakers. So I'm going to ask them to turn on their cameras now so that you can see them, and then I'll introduce them properly. There we go. We've got James on the screen. And there's David just appearing now. Brilliant. Hi, welcome to both of you. It's lovely to have you here today. Today we're first going to hear from David Hong, who's a senior lecturer in the Department of Chemistry at the University of Liverpool. David's research is focused on early stage drug discovery for infectious diseases, including malaria, tuberculosis, antibacterial infections and neglected tropical diseases. In addition to drug discovery, David has a strong interest in developing chemical probes for chemical biology and target identification, and in developing insecticides for vector control. And he was part of a team that won a 2024 prize, Horizon Prize. We're then going to hear from James Coverdale, who is a chartered chemist and assistant professor in pharmaceutical biology at the University of Birmingham, where his team are developing new mass spectrometry techniques to study the subcellular fate of catalytic anti-cancer metallodrugs in cells. James was part of the team awarded Royal Society of Chemistry Dalton Horizon Prize in 2022, and more recently has received the 2024 Royal Society of Chemistry Spectroscopy Award for his contribution to the field. So I'm very excited to hear from both of these speakers today. I'm sure you all are as well. Now, after we've heard from them, then don't forget we're going straight into the Q&A part of the webinar, so please do start sending in your questions for our speakers. But without further ado, we're going to hand over to David to begin. So we'll get his presentation shared on the screen now. Okay, there we go. I'll let you take it away.
Well, thank you very much, Natalie. Well, again, we would like to thank the Royal Society of Chemistry for this opportunity to present our work, which awarded the Horizon Prize for this year. And then today, I'm going to tell you the story for the quest of safe mycorrhizal drugs, the anti-Wolbachia drug discovery program. Before I get into our work, I do need to give you some background about the diseases that our drug is to treat. This involves two neglected tropical diseases caused by parasitic filarial worms. The first one is called onchocerciasis. We also know that as river blindness because the filarial worm infection actually caused the patient to go blind after a certain period of time. It affects more than 37 million people, mostly in the tropical region. The other one is called lymphatic filariasis. It also known as elephantiasis. The parasitic worm, they are mainly nestle, in this case, in the lymphatic system to cause the enlarge of the lymph of the patient, as you can see in those pictures. And the impacted area and the population, it's wider and bigger than onchocerciasis. It's affecting over 120 million people in the world and impacting quite a big area of subtropical countries. The diseases, they are transmitted by various factors, and it has a relatively complex life cycles. Both have different stages in the host and also in the vector as well. Currently, there is no vaccine or medication to prevent those infections, and the current treatment strategy is very lengthy. It will involve 2 to 3 drug combinations treated either annually or biannually from around 6 years to more than a decade. The main reason for such a lengthy treatment is because the current drug only targeting the microfilarial larvae of this worm, but not killing the adult worms. So the adult worm live for a very long time in the host, which leads to the long treatment. About 20 years ago, one of our team members, Professor Mark Taylor from the Liverpool School of Tropical Medicine, discovered that there is a bacteria that lives inside these parasitic worms they call Wolbachia. From this picture, you can see this is a slice of the worm. You can see the pain area is the Wolbachia that lives inside those parasitic worms. But more importantly, we discover Wolbachia, it's essential for larval development, embryogenesis, so the worm's capability to produce offspring, but also impacting adult worm survival. So if we can deplete Wolbachia from those worms, it will lead to a gradual death of the worm within 1 to 2 years. That will significantly reduce the treatment time and the recovery time, and without all the other side effects of rapidly killing of the adult worms. In fact, using doxycycline, a broad spectrum antibiotic, can deplete the Wolbachia inside those parasitic worms. So for example, after a 6 weeks treatment of doxycycline for this LF patient, and after 24 months, he makes a full recovery as shown in here. But the problem with the use of doxycycline, firstly, is the treatment regimen is too long, but also there is complication of contradiction to be used in young children or women at the childbearing age. So at the beginning of our program, a target product profile is being set up to find new anti-Wolbachia drugs for the treatment of these two diseases. Firstly, we want it to be much shorter, 7 days or less. It can be delivered orally and safe for children or women of childbearing age. Of course, facing the patient population the drug is being used, it is important that it will be compatible for the treatment of other tropical diseases like malaria or tuberculosis, and also the drug needs to be very cheap. So for such a complex program, it's quite obvious that we need a large team from different disciplines to work together to solve this problem. Today, I'm going to take you through 3 key aspects within this program and also demonstrate how different teams collaborate with each other to get to where we want it to be. The first part of it, it's involving compound library screening and cheminformatics to find our starting point. So at the very beginning of this program, about 15 years ago, we started from a relatively modest size library screening from a commercial source, about 10,000 compounds going through our primary screening, hit confirmation, hit validation, and then through a cheminformatic analysis, as shown in this picture, we identify 6 diverse hit series. I'm going to tell you a little bit more about one of the series later. Our story moving forward from that point, our later screening actually involving in a much larger library. At this point, an industrial partner, AstraZeneca, it's actually very kindly opened up their compound library, 1.3 million of them, to allow us to screen for anti-Wolbachia hits. At the same time, they also allow us to use their high throughput screening facility to enable us to increase our throughput from only be able to screen about 10,000 compounds in a year using manual method qPCR readout to screening 1.3 million compounds in 4 months, fully automated in high content imaging readout. The left-hand side figure shows the representation of those hits that identified in various stages of this process. And I will touch on the MF screen, the microfilarial screen, a little bit later on. But basically, we are not only using the cells in our screening process, but we also use the more appropriate subject, which is the worm itself, as the screen as well. Going through this process, we identify also a series of very high quality screening hit with diverse structure. They are also showing in this graph here. You can see different sphere now are representing different hits, and their structure are also representing in their relatively space in here, and the radar port is representing the hit molecules property. So from those radar port, you can see they do have some very nice property, but also they do have some weaknesses as well. So supporting by LifeArt at the moment, the AWOL team is currently taking some of this hit forward in the latest iteration of our optimization program. Maybe in a later day, I can tell you a bit more. But now I want to turn our attention to the medicinal chemistry and process development effort we're putting in before to talk about the series that I showed you before, the final pyrimidine series. Overall, the general synthetic strategy is quite modulated for this series of compounds. We can start it from a dichloral substitute intermediate through 2 selective aromatic substitution to install 2 different side chains into the 2 and 4 position of this pyrimidine core, and then end with some sort of functionalization of a different ring. To put this into context, this is our screening hit with relatively modest anti-Wolbachia activity at about 300 nanomolar and also quite a significant metabolic instability against both hepatocyte and microsome, we quickly move on to AWB124 that has an electron withdrawing group at the 4 position side chain to improve the anti-Wolbachia activity to 29 nanomolar, but at the same time also improve the metabolic stability significantly. At this point, we realize using the final pyrimidine core, we can only improve the metabolic stability to this point, because otherwise we'll start to impacting the potency. We use a scaffold hopping strategy to change the final pyrimidine core to a quinazoline core. Through this process, although we lost a little bit of anti-Wolbachia activity now to the 100 nanomolar range, but we gain a lot of metabolic stability, now presenting in the hepatocyte clearance and the microsome clearance. That's where we've been, where we want it to be. However, the physical chemical property, including solubility, it's being impacted in this process until we install another nitrogen into our core to make it into an azoquinazoline core, AWZ1010. We reinstall the potency against Wolbachia, but at the same time have a very balanced DMPK property as well. From this point on, we started to put this front runner into our worm model, but then very quickly realized there is a problem with this particular molecule. It's very well recognized by the PGP POM. PGP efflux normally would only impact oral absorption. However, in our case, because our filarial worms also have a very extensive PGP transporter systems that could actively pump out the drug that's been go in. So our drug, although looking very good against the cell, have significant problem against the worm in this case. So to do that, we also apply our medicinal chemistry strategies to methylate this nitrogen. This is the first magical methyl group that we see in action. We increase the anti-Wolbachia activity. At the same time, we really modulate the PGP efflux. However, we lost quite a bit of our metabolic stability until we cyclize this two to make this side chain at the 2 position a morphine ring with a methyl substitution at the 2 position. We gain good metabolic stability. We have reasonably good anti-Wolbachia activity and also modest efflux ratio. At the same time, we are seeing the second magic methyl effect in action as well, that with or without this methyl group not only impacting the aqueous solubility, but more importantly, is impacting our anti-Wolbachia activity in the worm too. So with AWZ1066 as our candidate, we have proved that it has the right efficacy, pharmacokinetic, and safety profile to match our target candidate profile. It can we can reduce Wolbachia in 7 days in comparison to doxycycline in 42 days in various animal models. I'm going to touch on the fast kill rate later on in the next part, but the other thing we find is AWZ1066. It's very specific against Wolbachia. Normally, for academic drug discovery program, the story would more or less end here. But very fortunately, we have an industrial partner, ESI, helping us to transform not only from the screening hit to a clinical candidate, but also take this into a process development using this three-stage process to produce more than 10 kilograms of the S isomer of the 1066 with high purity and good overall yield from the process. We optimize significantly of the two intermediate that required in this process and identify this hemi-HCl salt is the most stable form to formulate as the drug substance to the point that they can be made into tablet and go into clinical trial. So this is for a medicinal chemistry, what I would say, one of the most satisfactory outcome in this process. But of course, as I mentioned it before, there is this fast Q rate that we are observing in this process that to show that in comparison to other antibiotics, for example, doxycycline, in 1 or 2 days treatment as shown in this scheme, it works much better in comparison to any other antibiotics. That probably could explain why AWZ 1066 can work much faster comparing with, for example, doxycycline. But to understand the mechanism of action for phenotypic hit, it's a challenge. It is particularly a challenge for anti-Wolbachia drugs that it's a intracellular organism. So we have to come up with chemical probes that with various functional groups, for example, photoreactive groups or click handles, and use a chemical proteomics approach to identify potential target for this series of compound. The workflow is shown in this figure, starting from incubation with the Wolbachia infected cells, UV activation to covalently bind our probe with the protein of interest, then using click chemistry to bind biotin to enrich those protein and through mass spec analysis to get to a series of potential target of this. This is a work in collaboration with Imperial College and also Liverpool School of Tropical Medicine. What encouraged us even more, it's from our latest screening hit. We're looking at not only one, but a diverse set of fast-acting anti-Wolbachia compounds that have a similar sort of profile as AWZ 1066. What I'm taking you through very quickly in the last 20 minutes or so, it's this journey that we started from 2007. We only have one proof of concept drug in the form of doxycycline, to now in 2024, we have hundreds of hits. We take some of them into our lead optimization and progress them into clinical candidate. And some of those proof of concept drugs has now become WHO policy drugs that is being rolled out in the disease affecting region. So the hope is really lying ahead of us to hopefully to control these two neglected tropical diseases. And at the end, I would like to acknowledge all the contributions from a bigger team that is led by Steve Wall, Paul O'Neill, Fabian, and also Mark from various partners and the team photo showing here. And again, thanks for the Royal Society of Chemistry for giving us the horizon. Thank you.
Thank you so much, David. That was brilliant. What excellent work. It's really nice to have a little bit of detail there, and you've been able to take us through a really good introduction to that science. Thank you so much. We've actually got a couple of questions that have already come in, so I think I might ask you those now if that's all right, and then we'll expect to have some more at the end when we've heard from James as well. One of the questions that's come in is said, it asks, could an anti-Wolbachia approach actually eradicate river blindness, or does it just improve symptoms for those infected?
Well, it's a very good question. In comparison to the current drug treatment, anti-Wolbachia approach can actually, eradicate. It's a very specific term, but it can actually kill the adult worm. So it's not only relieve the symptoms, but also kill the adult worm. But unfortunately, if the patient is already blinded by the infection of the parasite, then that process is unreversible, unfortunately.
Right. Okay. Okay, well, I mean, that's brilliant news if that's not yet happened. And another question from James, it's quite a long one, so I'll just, I will read it out, otherwise if I try and summarise it, I'll probably lose the context. So they say there's a new patented technique openly available as open government level patents available from the Patent Office, where the experiments are performed under specific magnetic field as it greatly increases reactivity and yield. And they're asking, have you used these patented techniques? And if not so far, what do you believe the patented techniques can do for you and the world of drug discovery?
Well, this is a very interesting proposal that I am not aware of. So if possible, maybe we can connect after the webinar and talk a little bit more in depth into that. But I would say currently, no, we aren't using such technology yet.
Okay, no, that's fine. And yeah, James, if you want to have a look for David's email address online, it's freely available. Maybe pop him an email because that would be great to connect over that. Okay, right. I think what we'll do now is move on to James, and then David, thank you so much. We'll welcome you back at the end of the webinar, and we'll go through some more of the audience questions. So I'll welcome James on now. Thank you. You're back. We'll get your presentation shared, and we look forward to hearing more about your project. There we go. It's on screen now. Brilliant. I'll let you take it away. Thank you.
Lovely. Thank you so much. And as David said as well, thank you enormously to the Royal Society of Chemistry for not only giving us the chance to talk to you today, but also for the Horizon Prize that we were awarded back in 2022. So I am just one of the quite large team that were working on our project, which was looking at in-cell organometallic redox catalysis. When the work was first in its inception, I was actually a PhD student back at the University of Warwick. And as this work has evolved, I've gone from PhD student to postdoc to now running my own independent group at Birmingham. So it's something that I've always had this real passion for and driving behind. So I'm here just on behalf of our team today to talk you through some of our biggest findings. We're looking at targeting cancer specifically using a redox targeting approach. Now, for us, that means using metals, and we are very passionate about drug discovery beyond the world of carbon. There is a relatively established role of metals in medicine, and certainly their role in physiology is relatively well understood. As diagnostic agents, we see them very often in MRI and in X-ray, and for radiopharmaceuticals, of course. And the most common example that we talk about quite often is their use as therapeutics, and particularly as chemotherapeutics, like the platinum agents that are used very widely in the clinic. Now, we then think about how can we use this in a redox targeting approach? Because currently the pitfalls of therapeutics like cisplatin is that resistance is very commonly occurring, and that's something that we want to try and work around and avoid. We know that cancer cells are inherently sensitive to redox. Their metabolism is very different to that of a normal cell. They have dysfunctional mitochondria. And there is this inherent link between their metabolism and their redox. So we think about the key redox couples in cells that drive those processes like NAD, pyruvate lactate, glutathione. And these are some of the targets that I'm going to talk to you about in the examples from our work today. But we think hitting redox specifically is particularly advantageous because it is multi-targeting in its approach. It might avoid those pitfalls of a single targeting mechanism, which may incur resistance more readily. And we're thinking about also avoiding potential polypharmacology, so where drugs are used in combination, but often we then have to think about deleterious effects from drug-drug interactions that may be at play. So targeting redox in the cell as a whole for us can become a potential way to explore cancer treatments in the future. Our approach for doing this is to combine established catalyst chemistry that is very well understood in a chemical sense and in a chemical system, and thinking about how can we build that into the vast wealth of knowledge of metal drugs that have been developed for cancer. There are examples of ruthenium drugs that have reached clinical trials. Some of these are shown here, KP1019, NAMIA, and also then taking in the knowledge that, again, we have not only from the chemistry, but also from the biology of thinking about the challenges that we need to overcome. If we're going to go and do catalysis inside a cell, we have to bear in mind that high number of competing nucleophiles in cells, which are going to be possible poisons for my catalysts. And unlike a metalloenzyme, where that catalytic site is very hidden in that protein scaffold, in a small molecule catalyst, this would be quite exposed within the cell. So we really have to think about that balance between a catalyst that is active enough versus one that is stable enough to be functional in a biological system. And our approach to that is to think about second and third row transition metal centers. We're typically in the middle of the periodic table in the ruthenium, osmium, iridium, rhodium section. And we think about the use of half sandwich complexes quite a lot. So these are complexes with a metal core and a arene unit coordinated above the catalyst, which helps to stabilize that active catalyst in the oxidation state we want it to be in. So in a first example of this work, and where we really started to get some traction, was with this molecule shown in the top right of the screen. This is a molecule called FY26, developed by Peter Sadler's group, and works on the catalytic oxidation of NADH to NAD+. Now, it's worth saying that this drug by itself is already very potent, even if we don't necessarily have to worry about that redox targeting aspect. Looking at selectivity for cancer cells over non-cancerous cells is around 25 times. So we already have a very good potent selectivity. And looking into its mechanism in more detail, actually what we can see is that this molecule is capable of oxidizing NADH to NAD+, and tipping that redox balance inside the cell using molecular oxygen to carry out that oxidation. And by doing that, we generate hydrogen peroxide. We generate reactive oxygen species inside the cell as part of the mechanism of cell death. Now, this isn't necessarily new in the arena of metallodrugs. In quite a lot of examples, we do see ROS generation in cells, but this is one of the first times that we really started to understand that that generation was catalytic, and we can attribute a catalyst cycle to how that ROS is being generated, rather than it being an observational study. In this work, we also had some collaborative work with the synchrotron down in Grenoble, looking at localization of this drug inside the cell. So we actually tagged the ligand with a bromine atom, so we can look at both the metal center, the osmium, and the bromine at the same time within the same molecule, and start to get a flavor and a feel for where is this thing locating itself inside the cell? Where do I expect this catalytic mechanism to be occurring? From there, we then started to look in a bit more detail. We have some flow cytometry studies. So in the top left, these help us to understand how the type of reactive oxygen species that are being generated. And this biological data supported our chemical theory of an oxidation cycle involving the production of hydrogen peroxide. What we then were able to do in the lower left is in an in vivo model. This is in zebrafish embryos. We were able to show this dose correlating generation of reactive oxygen species, shown here in green, detected with a fluorescent probe. Going a step further from that, I guess the argument that we were having with ourselves is this ROS a secondary effect that we're seeing happening as a consequence of something else involving my metal catalyst, or is it something that is being generated directly? And one way we started to approach that problem, which I'm showing on the right-hand side, is to look at co-localization of my catalyst and the ROS. Are they happening in the same place that I would expect to see my drug? So in this case, my ROS fluorescence is shown in green, and the drug fluorescence, we made a fluorescently tagged drug molecule, is shown in red. And again, we see that overlap when we overlay those confocal microscopy images to see that actually, yes, the ROS is being generated in the same location that I would expect to see the catalyst. From there, we actually took this work a step further. We have a catalyst that we understand can generate reactive oxygen species inside the cell, a catalyst that we know is able to tip that redox balance as part of its mechanism of selectively targeting cancer cells. So on the very far left, we can show the potency difference between A2780, which is an ovarian cancer cell model, versus a healthy non-tumorigenic model, MRC5. And we've got a selectivity already of about 28 times. But going a step further, one thing we did know with this molecule is that there are interactions with glutathione, GSH. And so by co-administering this catalyst that generates redox with an inhibitor of GSH synthesis, we were actually able to further potentiate that potency even further, from 28 times actually up to almost 64 times potency difference between healthy cells and cancerous cells. And this really started to back up our first theories of the origin of the selectivity, this inherent redox selectivity of the cancer cell. This idea that a cell that is inherently vulnerable to a redox imbalance is that much easier to target than a healthy cell, although it has the same redox couples, perhaps isn't as sensitive to that tip. And therefore, that we believe this is what is reflected in the much lower potency in a healthy cell line. With this, we can actually go even further. And one thing we've been looking at more recently is looking at different types of molecules that are also redox modulators to have a combinatorial and synergistic approach as to how we would selectively target cancer. On the right is some examples using tetrathiamolybdate, which is a modulator of intracellular copper. Now, we know that copper in cells is inherently linked to controlling cellular redox chemistry, and actually by further co-administering cells with a non-toxic concentration of this modulator, alongside my catalyst, we get an even greater enhancement of that potency. So we've now got strategies where we have a drug that is redox targeting via oxidation. We've got ways to even further enhance that oxidation. But what about reduction? And at this point, we felt that we needed to look at perhaps the problem the other way up. At the moment, we've got some hypotheses of our catalytic mechanism. We've got some evidence, but actually there is this wealth of knowledge that metallic drugs generally just produce ROS. So can we look at this problem the other way up and instead look at reduction? And this was back in 2015. Our team reported the first example of an in-cell hydrogenation catalyst. This is a ruthenium catalyst that was able to be put into cells. It's stable in aqueous medium and was able to convert NAD+, this time reducing it to NADH. Now, of course, if I'm going to be doing a reduction, I need some source of reductant. And in this case, we used the molecule sodium formate. This chemistry stems from the work of Naori, which actually won the Nobel Prize back in 2001, where formic acid is used very routinely as a hydride source in the chemical industry. And actually, its sodium salt formate is relatively biological compatible. It's non-potent at millimolar concentrations. There is still some knowledge that we don't yet understand about its role in carbon metabolism, but for the purposes of this work, we're working with non-potent concentrations of that hydride source. And what we can see on the left-hand side is evidence that starts to support the mechanism that we expect. Formate that I've shown in blue can donate hydride. It can be that reductive source for a hydrogenation catalyst. And what we can see in this graph is that even if I administer a or a sublethal dose of my catalyst, by further increasing the concentration of formate, I start to get this what tends to suggest a catalytic mechanism of cell death. My catalyst's potency is enhanced by formate. To back up the mechanism of catalysis that we expect we're seeing, we swapped out formate for acetate, chemically very similar, differing only by a CH2 unit. But in this case, acetate cannot act as a hydride donor, and we don't see any difference in cell survival. With this work, there was actually a lot more in-depth study as well, that we were trying to convince ourselves that the catalyst is definitely inside the cell. So we did a lot of mass spectrometry to look at localization inside the cell. We looked at comparative elemental analysis of cells with or without formate treatment to really convince ourselves that it wasn't the case that formate was just allowing more of my metal drug into the cell. Actually, the levels of metallodrug are not affected at all by the concentration of formate that's administered. So we start to get a feeling that this effect is truly synergistic and is suggesting that we have this hydrogenation cycle going on. In green in the bottom right, this is actually measuring NAD/NADH ratios as we see that over time. So rather than looking at cell death, we're actually able to see that formate alone doesn't have any effect on that NAD/NADH ratio. Yet when we introduce my catalyst, that ratio starts to massively decrease. And again, this is something that we looked at over time, as well as in a chemical model system, as well as in cells. So at this point, and really at the halfway point of my talk, we've got this example of a ruthenium case that can do the reduction reaction of NAD+ to NADH. And I've got an oxidation catalyst that can take NADH and oxidize it to NAD. So depending on the choice of my catalyst, I can either do an oxidation or a reduction in cells. And the biological observations that we make alongside these measurements correspond exactly to what we would see. A reduction catalyst, we see a reduction in ROS levels, whereas with the oxidation catalyst, we see greater levels of reactive oxygen species. For our oxidative catalysts, we tend to see apoptosis, whereas we don't tend to see apoptosis being induced for our reductant catalysts. Now, this starts to become important if I think about how cisplatin works in cells. Its DNA binding and then triggering cellular apoptosis is one of the most widely accepted mechanisms. But perhaps in the case of cisplatin resistance, inducing apoptosis might not be the best idea for me. In the case of the mitochondrial membrane potential, so thinking about mitochondrial function, in the case of my oxidative catalysts in red, I do see a decrease in that potential, suggesting the involvement of mitochondria that may be at play. But that isn't necessarily the case in the case of my blue reduction catalyst, shown on the left-hand side. So at this point, we wondered, well, where do we go next? And really what I wanted to do as my contribution to this project was think about, we've got a redox couple here, NAD and NADH, which is completely symmetric. Can we do an asymmetric version of this inside cells? This time we moved to osmium. So rather than using a ruthenium catalyst, we dropped down a row of the periodic table. We gained a lot more stability of the catalyst, which was really important for us if we're going to drive high turnover numbers inside cells in that environment full of nucleophiles that could potentially deactivate my catalyst. And instead of looking at NAD and NADH, we then went to look at pyruvate and lactate and whether we could modulate these levels using an in-cell catalyst. Again, we're thinking about reduction, so we're thinking about using formate as our source of reducing hydride, effectively being irreversibly converted and oxidize itself to CO2. Once again, administering a sublethal dose of my catalyst and a non-toxic dose of formate, I see the same dose-dependent effect that I saw previously with ruthenium. In blue, as I increase formate, I see an increase in cell survival. This again is in ovarian cancer cells. And if I change formate for acetate, this effect is completely removed. So I start to have a feeling that my osmium chemistry is behaving somewhat similarly to the ruthenium chemistry, arguably actually with a slightly better reduction in cell survival relative to the ruthenium case, which we would suggest is partly attributed to the stability of that catalyst. What became more interesting is when we started to branch out and look at other types of cancer cells. So we looked at this effect in non-tumorigenic cells, shown in green. So in non-cancer cells, in what we would perhaps consider to be healthy cells, we don't see this effect. There is no potentiation by formate at all. This is similar to what we saw in the case of that oxidative catalyst previously, that in a cell that is very inherently vulnerable to that redox imbalance, this reduction by the catalyst and formate in combination is very selective towards cancer cells versus non-cancerous cells. We actually started to explore this even further by looking at cell lines which have high drug resistance, be that resistance to platinum drugs, adriamycin, triple negative breast cancer cells. So we're really thinking about those more difficult to treat cancers that patients really struggle with in the clinic, and we struggle with this treatment of those. But in that case, this catalytic mechanism maintains throughout. So we suggest that this is potentially a very powerful future treatment, thinking about those more difficult to treat cancers in the future. But we really, at this point, wanted some evidence that this is truly asymmetric, because at the moment we just have increased cell kill in cancer cells and not in a healthy cell, which is good. But of course, we're interested in the chemistry of these catalysts as well. So what we were able to do is we actually preferred to measure D-lactate, so the unnatural or the non-regularly occurring isomer of lactate in human cells. The advantage, of course, is that this is easily distinguishable from L-lactate if we use an enantioselective assay. So in the coloured bars in blue and red on the right-hand side, this is a chemical model system. So this is just putting some pyruvate into a tube with some catalyst and some formate. And depending on the chirality of my catalyst on that chiral diamine, I'm able to select for whether I'm going to produce L-lactate or D-lactate. And as we would expect, typically the EEs, so the enantiomeric excesses, are about the same depending on which way round you do the reaction. But this then gave us a really powerful advantage of moving this into cells, because perhaps if we were going to try and measure L-lactate production versus the background of L-lactate being produced in cancer cells by the Warburg effect, this isn't going to be easy to distinguish. So instead we picked D-lactate to detect, which is the figure on the right-hand side. And we see that there is a very clear increase of that when I have my catalyst and formate, that I'm able to selectively generate D-lactate in cells. And we speculate a possible transition state of what this is controlling this stereospecificity in the reduction. Finally, I just wanted to touch on what we've been doing since the Horizon Prize was awarded. And one of those things, amongst many other things, has been to look at whether we can further stabilize this catalyst. This is a more recent osmium example that we have reported, where you'll see on the right-hand side that that aromatic unit is now covalently tethered onto the other chiral diamine on the backbone. This actually builds on and is inspired by chemistry by Professor Martin Wills, who was part of our Horizon Prize team, who has commercialized in a chemical setting the ruthenium equivalents of these. So we moved to develop the osmium analogues, which we knew were particularly well suited for this in-cell catalysis. This was a really interesting system because the catalyst by itself, we do see that induction of ROS with the catalyst alone, which we suggest might be using something like NADH as the hydride source. We have some evidence to support that. And again, looking at those more difficult to treat breast cancer cell lines. So these are down the bottom. MCF7 are triple positive, whereas MDA and B231 are triple negative breast cancer, very difficult to treat. And yet that catalytic effect is relatively constant between the two versus the non-tumorigenic MCF10A cell line, where no effect once again is seen. I just wanted to finish by acknowledging, as I said at the start, that this is very much a team effort, and I'm just the representative for this much wider team between the universities of Warwick, Birmingham, Heriot-Watt, between our collaborators in France, in Switzerland, in China, in Germany. And thank you very much to the Royal Society of Chemistry again for the award of the Horizon Prize and for having us to talk today. Thank you.
Oh, thank you so much, James. That was that was a really detailed, brilliant overview. It's great work to see. Thank you so much for coming along to share that today. We have got questions coming in. Actually, before I get, David's coming back. That's absolutely fine. But before I come over to David, I've actually got a question directly for you, James. Let me just grab it from the questions box. It's a little bit busy there. Okay, I don't know if I'm going to say this right. After apoptosis, is that how you say that word? Okay.
Yeah, apoptosis.
Okay, good. It's not a word I've ever heard before, so that's a new one for me. I like learning new words. How do you expect the catalyst would be cleared from the body? Is there a risk of liver accumulation?
Potentially, so looking at biodistribution in an organism is not something we've been looking at massively yet. What we have seen in some of those XRF studies of cellular localization, at least, is that the catalyst seems to be broken down in lysosomes, and that tends to happen well before the 24-hour mark. So between kind of 12 to 24 hours. The metal itself is cleared a lot quicker than the catalyst itself, and that clearance of the metal seems to involve PGP. But how that looks in a whole organism, we're not as sure yet.
Okay, thank you. That's great. Okay, another question for you, James, and I've got one for David off that. What do you think is the greatest hurdle between the work you're doing and the clinical application of the work?
The fact that we aren't carbon. I think that is our biggest hurdle. I think there is a perception, and one I think that I had as well before working on this project, that somehow metals are bad. And with carbon, we have chemistry that we're very comfortable with. Mm-hmm. And I think there is a perception to be broken down, particularly around speciation, that if we think of cyanide versus sugar, well, they both contain carbon. And yet with metals, there is a very heavy generalization of all ruthenium complexes behave the same way, which we absolutely can see is not true. And I think that is probably one of our biggest hurdles to overcome, is that stereotype of what does a metal drug mean?
Yeah, I think you're absolutely right, because it makes you think of toxic things, and and so I think that's right. So it's more kind of that communication level and getting people comfortable with that. That's really interesting, actually. That's not something I would have thought of until you said that. But no, thank you so much. David, I've got a question for you. Thank you for coming back on. So the question is it possible or desirable to develop an environmental anti-Wolbachia pesticide to prevent infection in the first place?
Well, I think that's a very interesting notion that is being mentioned there. Interestingly, not only the filarial worm has Wolbachia in them, actually about 70% of insects also have Wolbachia as within them. So there is another Wolbachia program in the vector world that it's to look at modulating the level of Wolbachia inside mosquito to reduce the infection of, for example, malaria. And so far, from what I see, there isn't anything done related to when the filarial worm in the mosquito stage, what would happen if we can sort of deplete the Wolbachia at that stage. But I think that would be an interesting area to go into if possible.
Yeah, absolutely. Yeah, great idea. Thank you for that question. Another one for you, David. Are there any other applications for the anti-Wolbachia approach in relation to disease treatment or public health?
Well, apart from the one that I mentioned before, interestingly, not only human, but many other mammal, for example, companion animal like cats and dogs, they are able to host some form of filarial worms that has Wolbachia harboring in them. So there is actually some interest from the veterinary world that to use the anti-Wolbachia approach to treat the heartworms in cats and dogs. So that it's something that potentially it could also be used for as well.
Okay, thank you. That sounds great. Okay, a question for James from Ishmael. Osmium is a rare metal and it's relatively expensive. Won't that make your osmium systems expensive and difficult to translate into a commercial product? Great question.
Quite possibly, I think is my short answer to that. I can certainly vouch for the cost of developing these things because the material isn't particularly cheap. One thing that we are very interested in now is actually moving around that space in the periodic table. So we have systems that are relatively well established in ruthenium and in osmium and in iridium, which of course are famously not the cheapest. So now we have a better understanding of, and actually with the future work I was talking about with that covalent tethering between all of those ligands, we are now thinking about whether there are strategies that we can try and, as I would put it, push up in the periodic table to try and think about whether we can translate the system to more readily abundant and perhaps cheaper element centers, but still maintaining that catalytic effect.
Yeah, absolutely. So yeah, I guess that's the point of this research, right? You do the research and actually you're not initially worrying about those costs and those bigger picture things because you're just trying to get the result. And then actually, once you know you can do something, it's then reaching out and continuing that work. Actually, leading on from that, I'm interested now, obviously this work's been recognised and it's a big project. You're working across the world, it's a big team. I've got a question about that. How does that work? Do you find partly when people move institutions, is that a problem? Does the work continue? Do you find it hard to bring all these different people together working on such a big project? And then my second part of that question is what's next? What are you going to be planning next for this piece of work? I think you've already mentioned some of those things, which we've highlighted just there.
Yeah, that's a great question. I can speak from my own experience of this team of jumping, well, actually around the pandemic, I jumped into industry for a bit, I jumped into the NHS for a bit. For me, I'm very lucky to be surrounded by very passionate people who really, their heart and soul is in this project. And I think the same effect rubbed off on me. So as I've been moving around different institutions, that's only ever been a benefit to this project of gaining more experience and gaining access to new methods and new knowledge by moving institutions. And actually, to be honest, I would say that's probably one of the biggest strengths of our team, that we are all very flexible. This work is inherently, of course, very chemistry, but there is a strong amount of biology in that. There is a lot of computational chemistry, there's a lot of physical chemistry. And so we have all, as a team, had to be very adaptive to that as well, be that traveling around the world to work in different labs or just in our own approach in our own labs of how we work. And the other half of the question about what's coming next.
That's a big question.
Yes, we are still incredibly excited. And I think one of the biggest things that has always sat with us is that we only are really measuring what we look for. And certainly in biology and in inorganic chemistry, this is very true. We have a catalyst that we are kind of extending and saying, well, it's redox targeting. And really, that is such a big question in itself. We've looked at some representative examples in NAD and in pyruvate lactate. But really, what we're trying to get a bigger feel of is more generally, how does that look? So the project is moving at the moment into a lot more pharmacogenomics and transcriptomics, and really trying to look at the cell as a bigger picture of how is this catalyst system behaving in a more biological sense. And again, this is where our team has had to evolve our own knowledge, bringing new expertise in order to answer those questions.
Yeah, absolutely. Thank you so much, David. I'm going to ask you a similar question, that kind of the last part of that question for you, because we are running short on time, but I'd love to hear kind of what are the next steps for your work and the project as a whole.
Well, as I mentioned before, those screening hit from the AstraZeneca library, we are supported by Life Art funding. Together as a team, we are taking those forward as the next generation of anti-Wolbachia drugs. That's one thing. And also, in a way related to that, we have actually accumulated quite a large amount of data that associate with chemical structures and then anti-Wolbachia activities. So we are now pushing towards the model building using machine learning to mine those data to help us to build up something that potentially in the future we could maybe generate some sort of model that could do virtual screening instead of physical screening to find other hits in the future when it's necessary, or sort of use those learning for other neglected tropical diseases as well. So those are the sort of current work that we are investigating.
Wow, that's brilliant. Thank you. Yeah, it's very wide reaching, isn't it? Thank you so much to both of you. I'm afraid we are out of time now. So I'm very sorry if I didn't get to your question in the questions box, but please do contact the speakers if you feel like it's something you would like to follow up on. I'm sure they'd be more than happy to hear from you. But thank you so much to David and James for joining us today to share their science, and we can do a little celebration of their prizes as well. And thank you to everyone watching. You've got involved in the conversation. That's been great. We love to hear from you, as always, with all of our webinars. Just a reminder, the RSC's Horizon Prizes are currently open for nominations and will close on the 14th of January in 2025. The prizes, as we've spoken, are awarded to groups, teams and collaborations of any form or size and from any sector, and they recognise discoveries and breakthroughs and advances in the chemical sciences. The important thing is that each and every individual who contributes to the prize winning work is recognised as part of the prize, which is really nice, and it's very, very unusual to see that. So please do check out that page, which we've shared a link for in the chat. But I'm afraid that is all we have time for today. So from everyone here at Chemistry World, we look forward to seeing you again in a future Chemistry World webinar. Thank you and goodbye.